talk about ribulose 1,5-bisphosphate carboxylase oxygenase, otherwise known as Rubisco. Rubisco is a large protein of about 500,000 daltons. It has a notable axis of symmetry right here, but as we'll see later, there are several axes of symmetry. There are many prominent alpha helices and beta sheets that play an important role. Rubisco is the most important enzyme on Earth. It's present in all terrestrial and marine plants, eukaryotic algae, cyanobacteria, and other types of autotrophic bacteria. Here's an example: spinach. Rubisco in spinach and other plants is almost identical, but their sequences have some differences. Here we compare the sequence for spinach and the sequence for Arabidopsis, which is a kind of watercress. So at the top, the 1AA1 is the sequence for spinach, and 4W5W is the sequence for Arabidopsis. All of these green amino acids are the ones that exactly match between both sequences. The pink ones are similar amino acids. For example, the S and the T play similar roles. And then blue amino acids don't match at all. The brown amino acids are insertions or deletions. Sometimes we have a large chunk in one that doesn't all exist in the other. So the total number of amino acids is very different. But as you can see, there are many similarities between both sequences. Rubisco has some interesting characteristics that sets it apart from most other enzymes. For example, it's very slow. Well, while most enzymes react at a rate of about 1,000 substrates a second, Rubisco can only process 3 carbon dioxide molecules per second. To compensate for this, plants produce a huge amount of Rubisco. They have about half the space in each cell is taken up by chloroplasts full of Rubisco. Another reason for Rubisco being inefficient is that it will bind oxygen just as well as it will bind carbon dioxide, meaning that when oxygen does bind, it has to go through an expensive energetic process in order to get rid of the oxygen and replace it with carbon dioxide. Overall, Rubisco looks like a hollow barrel with caps on each end. It has a central solvent channel running through the middle, which defines a 4-fold axis of symmetry. The subunits are linked together by disulfide bonds. There are 16 subunits in Rubisco, making it a heterohexadecamer. 8 large subunits arranged as a tetramer of dimers, forming the sides of the barrel, and 8 small subunits arranged as tetramers at each of the poles. The large subunit originates from the RBCL gene in the chloroplast. All 8 large subunits are identical, with approximately 475 amino acids each, depending on the organism. An important cofactor for the large subunit is magnesium, and each subunit binds just one magnesium ion. The L subunit has many alpha helices and beta sheets, forming an alpha-beta barrel, which is where the magnesium ion lies, and it constructs the active site. The small subunit comes from the RBCS gene, also originating in the chloroplast. All 8 small subunits are identical, and they consist of approximately 123 amino acids. 2 important chaperones for the formation of Rubisco are the Rubisco accumulation factor 1, or RAF1, and the RBCX. They're kind of redundant, but they're important in order for Rubisco production to be efficient. RAF1 assists in the formation of the large subunit, and then RBCX also helps to form the large subunit dimer, and then forms the entire conglomeration of large subunits. When the S subunit comes along to bind the L subunits, the RBCX and RAF1 are ejected from the overall structure. There are about 10 active site residues. As you can see, there are about 4 lysines The magnesium binds one of the lysines down here as well, and it is stabilized by 2 other amino acid residues next to it. In the middle we have the RUBP, which is the substrate for Rubisco, and the carbon dioxide. They are held together by both being bound to magnesium, and only after being stabilized there do they react with each other. So the first step in the Rubisco mechanism is the conversion of RUBP to an enediolate by binding the carbamate group on lysine 201. Carbon dioxide then also binds the magnesium, displacing a water molecule that used to be there. And the metal ion causes the carbon dioxide to be polarized, which allows the carbon dioxide to perform an electrophilic attack on the enediolate. Hydrogen atoms are rearranged through several steps, resulting in a 6-carbon sugar. Then several hydroxylation and proton transfer steps are followed by cleavage, producing 2 molecules of D3-phosphoglycerate. The active site is the smallest functional unit of Rubisco is a dimer of 2 L subunits, and the active site is located at the interface of 2 L subunits which are parallel to each other. The small subunit has loops near the pore leading to the central solvent channel. Mutations in the beta strands forming these loops can cause changes in reaction rates, specificity, and denaturation. Some bacteria don't even have the S subunit because it's not involved in the actual reaction mechanism, as we'll see soon. So here we have the spinach Rubisco. It's the barrel consisting of large subunits and then caps on both ends of the barrel. For many photosynthetic bacteria, instead of having the barrel structure, we only see a dimer of large subunits. It doesn't have any small subunits because the dimer is enough for the reaction to take place.
So Rubisco catalyzes a reaction that's involved in carbon fixation, which is part of the Calvin cycle. It's the phase 1 of the Calvin cycle. The Calvin cycle is where water, CO2, and light are used to make sugar and oxygen. So the substrates for Rubisco are CO2 and ribulose 1,5-bisphosphate, which is RUBP. The products are 2,3-phosphoglycerates. As you can see in this diagram, CO2 and RUBP come together at Rubisco, and the RUBP is cleaved to form the 2 phosphoglycerates. This is the Calvin cycle. As you can see, phase 1 is carbon fixation. That's where the Rubisco is. Carbon dioxide comes in, as well as the RUBP. And then 1 phosphoglycerate is used throughout the rest of the cycle. It's transformed to reproduce the RUBP. And then 1 is released into the central metabolic pathways. So, as I said before, RUBP is one of the substrates that's used to make the phosphoglycerate. 1 phosphoglycerate is released into the central metabolic pathways, which is then used to synthesize larger molecules such as glucose. A naturally occurring inhibitor is the CAP, or 2-carboxylarabinitol 1,5-bisphosphate. As you can see, it's very similar to the intermediate of RUBP, which is right here, and this is the CAP. The only difference is the 1 oxygen that's added. So it's very similar, and it acts as a competitive inhibitor and won't allow the RUBP to continue through the reaction. Antigenic determinants are also known as epitopes. This is where the antibody can bind onto the protein. These are 3 examples of each. So we have RBCL, which is the large subunit above, and these are 3 epitopes there, and then 3 epitopes at the RBCS subunits. This plays an important role in immunoblot analysis. So antibodies are used to attach and to show where, if specific proteins are found in a sample. So knowing where the epitopes are is very important in this. So the primary literature that we chose is called Improving Recombinant Rubisco Biogenesis, Plant Photosynthesis and Growth by Co-Expressing its Ancillary RAF1 Chaperone. So recently, many researchers have been focusing on improving photosynthesis via Rubisco. Since Rubisco is very inefficient and slow, changing Rubisco or enhancing it is a good way to improve photosynthesis, which means greater crop yield, which can solve hunger problems throughout the world. RAF1 played an important role in this research, chaperone that assists in the assembly of the L subunit, and Arabidopsis also played an important role in this research. They used the Arabidopsis gene to see if RAF1 and the gene for the large subunit in tobacco plant could be enhanced using genes from the Arabidopsis plant. So the methods, the first thing they did was a bioinformatics analysis. They created a phylogenetic tree to compare the genes for RAF1 and the RBCL using a BLAST algorithm. This was to see if the two genes co-evolved together. The next thing they did was the plastome transformation. So a plastome is the DNA found in the chloroplast. So first they amplified, using PCR, the RBCL gene, and then they cloned the RAF1 gene downstream of this and inserted it into the plant. Plants were grown at 0.5% volume per volume carbon dioxide and fertilized with wild-type pollen. The progeny of these plants were then used in the research and to test the protein and the RNA content. So after they grew these, they tested the RNA and protein content using the ND page, SDS page, and immunoblot analysis. They also used a northern blot analysis for the RNA, the RNA content. So this is a diagram from the research. A is the plastomes they used. So they have the wild type initial DNA sequence, and then as you can see, they added the Arabidopsis RBCL gene and the Arabidopsis RAF1 gene. B shows the northern blot that they got, which is the RNA content, and then C and D shows the protein content that they got from the SDS and ND page analysis. After this, they quantified the catalysis of Rubisco. So they found the rate of the Rubisco fixation, turnover rate, and Rubisco specificity. They also measured the photosynthesis rates, and then they ran an immunoblot analysis to further study the protein content. And then, as stated before, they ran the ND page, SDS page, and immunoblot analysis. The results showed, initially, the phylogenetic tree showed that there was a coevolution between the RAF1 and RBCL genes. Because they co-evolved together, that meant they only were able to work together. So the plants that only had the RAF1 gene from the Arabidopsis plant, but not the RBCL gene from the Arabidopsis plant, did not show improvement at all. But the plants that had both the co-evolved RAF1 and RBCL gene from the Arabidopsis plant and neither gene from the tobacco plant showed improved affinity for CO2. The RAF1 chaperone was shown to successfully and stably form the dimer. Photosynthesis and plant growth both increased in the treated plant, and there were also higher levels and quicker production of Rubisco in the treated plants. Thank you. That was our presentation on Rubisco.